Hi, good afternoon, everyone. On behalf of Voice of Healthcare and Tata Medical and Diagnostics, I would like to welcome all the attendees and speakers for this webinar that is brought to you by VOH in collaboration with Tata MD. The topic of this webinar is Signs of Variance and Challenges in Variance Detection. We have a set of eminent experts here who would be expressing their views on the topic, and leading this interaction would be Dr. V. Ravi. Dr. Ravi is a renowned virologist and head R&D at Tata Medical and Diagnostics. Further to this, I would now like to hand over to Dr. Ravi, who would be leading this conversation. Over to you, sir.
Thank you, Devjani, and namaskar, and welcome to all of you. This afternoon, we are having a 1 hour session. The first part of it is panel discussion on the challenges posed by the variants of SARS-CoV-2. And in the last 2 years since SARS-CoV-2 was discovered, we have had many upheavals. It has been a roller coaster ride. Each time people expecting that the pandemic is going to get over. And just 4 weeks ago, we had the news that we have another variant which is likely to trigger off a third wave. So it's on this background that Tata MD decided to have a knowledge session, also to introduce some of the solutions Tata MD has come up with for Express PCR Solutions, as it is called. I have with me a very distinguished panel, and I would like to introduce them. First, my dear friend, Professor Ashok Rattan. Professor Rattan and I go back a long way, 32 years to be precise, since I met him, and we've been together through our journeys in microbiology. He is an advisor for Quality Research and Development Forum, Pathkind Laboratories, and he's also WHO Lab Director for CAREC PAHO. Welcome, Dr. Ashok Rattan. The second expert we have today is Dr. Chaitali Nikam, Head Infectious Disease and Research, Thyrocare Technologies Limited. Welcome, Dr. Chaitali. And the third expert we have today is Dr. Devjani, Head of Microbiology and Serology, Dr. Dang's laboratory, and she also is a member of the Executive Committee of Indian Association of Medical Microbiologists. So the way we have organized this panel discussion is we have 5 burning questions which will be addressed to each of the panelists, and they'll give their responses. So without wasting time, let me shoot the first question. To you, Professor Ashok Rattan, followed by Dr. Chaitali, and then Dr. Devjani. Why do SARS-CoV-2 variants keep emerging? I think this is a question that is there on everybody's mind.
Thank you, Professor Ravi. Genomes of RNA viruses like hepatitis C, HIV, and influenza are usually of 10 kilobases length and have no proofreading enzyme activity. While therefore the multiplication is highly error prone. Genome of SARS-CoV-2, on the other hand, is 3 times as long, and there is an enzyme NSP14 exon N with proofreading activity. SARS-CoV-2 accumulates about 1 to 2 mutations every month. Even though there is this proofreading activity, this mutation accumulation is half of that of influenza and 1/4 of that of HIV. Mutation is a change in one single nucleotide in the genome, in the genetic code. Mutation is called a variant when one or more mutation confers a distinct property on the offsprings of the virus, and this could be interference with diagnostic tests, decreased susceptibility to one or more class of therapy, decrease in neutralization by antibodies produced as a consequence of recovery from infection or vaccination, evidence of increase in transmission, or evidence of increase in severity. In SARS-CoV-2, 4 classes of variants are being monitored. One is called as variants being monitored, which is there are 10 variants being monitored from alpha to zeta. Then there is variant of interest. At present, there is no variant of interest being monitored. Then there are variants of concern, which includes Delta and now Omicron. And then there is fourth will be variant of high consequence. At present, there are no variants of high consequence.
So
Thank
just
you,
to
Professor
add,
Rattan. Dr. Chaitali, please.
yeah. So just to add up with Dr. Rattan, so basically variants emerged when viruses mutate, and viruses are constantly experience the changes or mutation to their genetic material when they replicate their RNA or genetic makeup when in the host cells. So this is actually a part of pathogen's life cycle and helps it to survive. And most of these are inconsequential, means they may get sometimes by their own. So as variant is having one or more mutations, that is differentiated it from other variants of SARS-CoV-2. And as expected, multiple variants of SARS-CoV-2 have been documented globally throughout this pandemic. And this is the reason that it is, gets changes and experience lots of changes in the survival for them. So
Thank
this
you,
is from
Dr.
just addition to Dr. Rattan from my end.
Thank you. Dr. Devjan?
So this is a very pertinent question, more so today as India sees a surge in Omicron cases to 271 as we speak. And mutation is a mechanism for the virus to evolve in order to have a survival advantage. So mutations not always are consequential, some may be inconsequential, some may be detrimental to the virus itself, but sometimes variants arise. So when they accumulate one or more mutations, variants are then classified, as Dr. Rattan said, into variant under investigation, variant of concern, variant of high consequence, and so on. So to keep the variant classification simple, WHO has given a very simple nomenclature for which destigmatizes as well as which is easy to use for communication. So that is about variants. And most of our knowledge about RNA viruses comes from influenza virus. Now, influenza virus doesn't have a proofreading mechanism. Whereas SARS-CoV-2 does have a proofreading mechanism. So it is seen that the mutation rate is about 4x lower as compared to influenza virus.
Thank you, Dr. Devjan. Moving on to the next question that generally people ask in the context of variances. What are the challenges in the rapid detection of SARS-CoV-2 variants? You heard Professor Rattan and others also mentioned that variants can impact diagnosis. So what are the challenges in the rapid detection? Professor Ashok Rattan, please.
The gold standard test which has been used for detection of SARS-CoV-2 is real-time Rtpcr. This has focused on various genes, the N gene, the S gene, the E gene, the RDRP gene, or ORF1ab gene. Some of the tests are single gene and some of them are multiple gene. If the test is that of single gene and the mutation is present in the site where there's binding of the primer, then there'll be silencing, which occurred once in Nelly, that is the alpha gene, that alpha mutation that occurred. There was deletion of position 6970, which was on the spike protein, which was the site for the attachment of the primer for the S gene. So this S gene silencing. Now we are noticing N gene silencing. So whenever there is a mutation, and that is mutation at the site of binding of the primers, there will be changes. And that is why it's been suggested that instead of single gene, we should have multiple genes for detection for virus.
Dr. Chaitali?
Yeah, so as Dr. Rattan rightly mentioned, all the aspect about challenges. So in the line of that, most of the molecular tests for SARS-CoV-2 are designed to detect specific RNA sequence found in the viral genome. However, the SARS-CoV-2 virus mutates regularly, as we discussed just now, in several genetically unique variants, and each with the different RNA sequences that may be found in infected individuals as the pandemic progresses. So since molecular tests took for a specific sequence, it is possible for certain viruses to mutations to affect the performance of certain tests if the mutation is region of the genome targeted by that particular kit. As Dr. Rattan rightly said that there are specific kits for specific regions like RDRP, N gene and S gene, and the clinical impact of genetic variants on this test sensitivity is largely influenced by the sequence of the variant. So this is the first challenge which I feel. Second one is the design of the test and the prevalence. And third one is the prevalence of the variant in the particular population. At the end, I would like to highlight the same thing which Dr. Rattan has highlighted, that tests that rely on the detection of a multiple region of the genome may be less impacted by genetic variation in SARS-CoV-2 genome than the tests that rely on the detection of only single region. That is from my end.
Thank you, Dr. Chaitali. Devjani?
So, sir, in order to answer that, the challenges in rapid detection of variants, I would want to divide it into two. Firstly, the current testing algorithm for variants per se is that all samples undergo a molecular testing for SARS-CoV-2, which is the generic test for all SARS-CoV-2, including variants. The step two is, of course, the pending confirmation for genome sequencing. So the challenges could be at both steps. Firstly, the challenge in the molecular assay. WHO has always stressed that you use dual target or multiplex assays and not just rely on a monoplex assay, because this could give you a false negative. And you should be very cautious about false negatives if there are variants in circulation, and especially if the pre-test probability is high. Secondly, coming to genome sequencing, what are the challenges? Because that is the gold standard currently. So it is time consuming, labor intensive, it is expensive. Currently under INSACOG, about 38 centers are doing it, which includes 10 central and 28 regional centers. So the outreach to periphery, to rural areas, it would take time for logistics. And also it is CT value dependent. So samples which have more than 25 CT value would not have a very good sequencing performance. So these are the limitations per se for confirmation of variants currently.
Thank you, Devjani. Now moving on to the third question. What are the challenges you have mentioned already? What are the current tests that are used for SARS-CoV-2 detection? Do they detect Omicron variant? Or other way is asking is, do they detect all the variants that one is currently concerned about? Let's say Delta, which is present along with Omicron. So what is your opinion? You already said how variation can impact. So what do you think are the tests currently available? Will they answer? And also a word about probably antigen detection tests, because that is also still being used. Professor Ashok.
There are, with the Omicron coming into picture, those tests which depended only on N gene, single gene test like Meridian Biosciences, Tide Laboratories, Applied DNA Science, Lina COVID-19 assay, these depended only on N gene, they would fail. so that Omicron will not be detected by these tests. On the other hand, if we are using a multi-gene target, multi-gene assay like TAGPath or Eldona, then there will be S-gene silencing. When that occurs, that will give you a hint that maybe here now, since there is S-gene target failure, that would mean that there is a mutation in the S gene, and this may be Omicron again. So that is what you would be looking at. At the same time, Indian companies have been working on Delta, and now it appears that GenePath from Pune has been able to develop a test which detects both Delta and Omicron at the same time. They have, of course, multiple genes, and it's only when you use multiple genes would you be able to, one, screen, and second, detect.
Thank you. Dr. Chaitali?
So currently COVID-19 tests which are in the market and having ICMR approval and all those things. So as Dr. Rattan said that, TAGPath from or S-gene dropout cases can give us the hint. But currently COVID-19 tests are designed and authorized to check broadly for SARS-CoV-2 virus and not specifically for any VOC or VOI. And it is common for all viruses to change and mutate over time and resulting in different virus strains. Also, as per literature, which says that there are tests in FDA pipeline for specific detection of a variant. And to be precise, these tests are identified based on initial bioinformatics analysis, which has been done by the bioinformatics platform, which are available online. And these tests are not yet have been evaluated against the Omicron variant or any VOC in the laboratory. So, so far, the tests which are in pipeline are also not authorized to determine the presence or absence of any VOC or VOI for that matter. That's what I think.
Thank you. Dr. Divjan?
So the impact of a variant on a test would depend on three factors. First is the sequence of the variant, second is the test design, and third also the prevalence of the variant. So as we know, the Omicron has gathered so many mutations, and so many unique mutations are there on the spike protein itself, almost 26. And so this could have a potential impact. Now, FDA on the 23rd of September has issued fresh guidelines for emergency use authorizations, whereby there have been additional conditions of authorization in response to the continued emergence of variants. So in order to do that, every test developer needs to submit additional data of how the test being developed by the company is compares with the variants, how it what's the test performance. So and as Dr. Rattan has already said, there are 3 kids that have been in a recent FDA communication which have been withheld until the issue is resolved. These are mostly monoplex assays. 2 of them are N-gene based assays, and the other one has 2 targets, but both of them are on the S gene, which has unfortunately undergone the mutation. So, but we don't have any such communication from ICMR regarding our kids. Coming to rapid antigen test, there have been 4 mutations in the N gene, which is the primary target for rapid antigen test. However, it doesn't seem to impact the test, but there could be certain caveats that have to be looked into as further evidence appears.
Thank you. Thank you, 3 experts. The sum and substance of what you're saying is, as long as multiple genes are used as targets in real-time PCR, and they are designed in a way that they cover conserved regions of the genome, these tests are good enough to pick up Omicron. They will not miss Omicron, not pick up Omicron. They will not miss Omicron. My next question is, there's a lot of buzz in the last few days, ever since the emergence of Omicron, on spike gene target failure, SGTF as an acronym that is being used, which very nicely all of you explained that there will be a dropout of the spike gene, and that can be used as an advantage to suspect Omicron in a sample. So I want a little information on how reliable it is. Is it foolproof evidence? What do you think is the data that is emerging on the spike gene target failure assays? Over. Professor Rattan.
There is, of course, I think sequencing is the ultimate to identify the variant. The S gene or N gene dropout would only make you suspect and make you aware that something is happening. Recently, Pune-based GenePath has developed a new kit called 𝐬𝐶 Delta, which can simultaneously detect Delta and Omicron with 100% sensitivity and specificity. This kit targets multiple virus-specific genes and detects all lineage of Delta and Omicron. There is also ICMR Regional Medical Research Center, Dibrugarh, which has developed a hydrolysis probe-based real-time Rtpcr for detection of Omicron in 2 hours under Dr. Biswajyoti Barkakoti leadership. This kit will be manufactured by GCC Biotech of Calcutta under PPP model. And Dibrugarh was the third lab which could isolate Omicron, and after that they claim that their test is 100% sensitive and 100% specific for detection of Omicron. But ultimately, I do agree that it is sequencing which will determine. Though I feel that nanopore sequencing is error-prone, so you cannot be looking for mutations in an error-prone sequencing sequence.
Thank you, Professor Ashok. Dr. Chaitali.
Yeah, so just to highlight few of the point which has been mentioned by Dr. Rattan already. So to the basics, the target failure which has been, it's going so far, that is S gene dropout or N gene dropout. So all this dropout may occur due to different mutation in other variant and may not be specific to the Omicron variant or any VOC. So target failure may also be observed without the presence of a mutation in some time in the targeted area due to the sensitivity of genetic target. So therefore, the presence of a target failure detection pattern is definitely having some value, but it's not 100% definitive for confirmation of the presence of the Omicron variant. So for example, if we are taking here one generic target is expected to have a significantly reduced sensitivity due to a mutation in SARS-CoV-2 Omicron variant, which is B112.529. So it is a specific deletion in the spike region of 6970. So which result in S gene dropout and also referred as a S gene dropout failure assays, and where the genetic target that covers the portion of the S gene where the deletion occurs failed to detect the virus. So it may happen in other cases also besides Omicron. Also, there is a data which says that the N gene and S gene dropout are typically not observed in the Delta variant, which was high during our second COVID wave, and specimens with the target failure detection pattern may Omicron variant and should be prioritized for sequencing confirmation. Also, the presence of target failure detection pattern just signalize that sequence should be considered or prioritized for such samples where we can see the S gene dropout, but eventually it may not replace sequencing. That's what I feel.
Thank you very much. Dr. Devjani?
So, sir, this pattern can be picked up to enhance the surveillance activities. However, it is not foolproof because there is emergence of a lineage, which is BA.2. So here you don't see the SGTF happening because there is the deletion in 6970 position is not there. So you could miss if you just rely on a SGTF assay. So pending sequencing still remains confirmatory. So the caution has to be high. You have to take the travel history, your contact history, everything else into account before you report out any result. So that is the limitation of SGTF assay being used as the only proxy marker. Having said that, SGTF could be used for, especially for at triage at airports, where because there is a lot of burden on international traveler reports who are waiting for their sequencing results. So these assays could kind of allay anxiety and the stress, the burden on sequencing labs per se.
Thank you very much. All of you have said very clearly that the S-gene dropout or N-gene dropout assays are good surrogate markers, but they're not confirmatory. So that brings us to the last question in the panel discussion. Sequencing is the only method of confirmation. What are the challenges in sequencing? And do you think there could be alternatives or solutions to these challenges in sequencing? Professor Ashok Rattan?
Yes, I think as we agree that sequencing of the S gene will be the best, but maybe multiplex PCR for different regions so that the 1273 base pair that we have to sequence could be done quickly by multiplex PCR and look for any mutations in that might be one way out, so that the results are available in a few hours rather than 4 to 5 days that it's taking. The second one has been a request from most of the private labs that they would also like to participate in sequencing activity, because I think their turnaround time is always faster than. And I remember we had one of our colleagues suffer from Delta variant infection in March, and we did submit a sample, but we still haven't got the report yet. So I guess if you get private players into the act, then it'll be faster and we will make sure that the report reaches the right person.
Thank you. Dr. Chaitali?
So a diagnostic platform for variant detection on sequencing and are suitable for the use at point of care, such as core-based technologies, are anticipated to contribute substantially for variant detection process in near future, and being capable of diagnosing variant calling or genealogy and novel mutation detection. Also, as an alternatively, there are a few Rtpcr-based SARS-CoV-2 variant identification panel, which is under research use only, that is RUO, from some of the companies are already in market and in pipeline of WHO or FDA for known variants and mutation. It's very clear that the variants which is already analyzed on the based on the mutations can be tracked by this new Rtpcr-based RT-PCR-based test, which are coming up in pipeline. But as Dr. Rattan said, definitely only S-gene sequencing for partial S-gene can be also an alternative to speed up the procedure for genome sequencing than doing whole genome sequencing when we know there is S-gene dropout. That's from my answer.
Thank you very much. And Dr. Devjani?
Most of it has been covered by my co-panelists very well. So variant specific RT-PCR could be the solution to escalate the surveillance activities. Now, there are various assays which are coming up, like the Joint Research Commission, European Joint Research Commission has developed the OMET, which targets specific sequences to the Omicron, and there's no cross-reactivity with other variants. There are Indian kits which are being developed, the 𝐎𝐿-Bio, which they have a kit which is ready to go if the need arises. And the good thing about 𝐎𝐿-Bio, the TrueNat machine is widely available. So there are 4,000 equipments placed across the country, which can ramp up, testing to approximately 4 lakhs per day. So that it is a very good way of scaling up. If we want to detect Omicron, then there is a kit from IIT which they have developed, and they are filing an Indian patent for the same. However, having said that, detect Omicron specific PCRs should only be used as a second line. So the first generic assay should be your SARS-CoV-2 generic PCR, and the variant specific should be the second line. Otherwise, there will be development of a bias and you would miss out the wild types or the other variants.
Thank you all very much for a very enriching panel discussion, for informing the audience on the science of variants and the challenges in variant detection. We hope that Omicron will not, for the good of everybody, will not cause as big a wave as the second wave. But whatever indications we have, it looks like it is going to cause a third wave. So let's all brace up for it and continue our efforts in helping diagnosis as microbiologists, and also contribute in a way, hopefully, for development of assays that can, raise to the challenge and help in diagnosing and alleviating the suffering of people. Thank you very much, all of you. I appreciate and value the time that you have spent with us. Thank you.
Thank you.
Thank you.
Thank you.
Thank you all the panelists for setting up this beautiful panel discussion. We have a few questions in the chat box and the Q&A section. I would now request Sachin to please take up a few questions for our speakers to address.
Thank you, Khushbu. Thank you to the panel. So, Dr. Ravi, it seems that there are still a couple of questions on, the S and N dropout, with a specific question from Dr. Kavita, where she is asking that with S dropout and N dropout, if the kit contains only N and S gene, can there be a chance of missing out an Omicron infection?
May I request Professor Rattan?
Yes, of course. If the test is only single gene and there is mutation, there is a nine nucleotide mutations in the N gene, then, as I said, in Tide as well as in Meridian, the kit, it will report negative. So Omicron will be missed.
I just want to add to that. During the second wave, as one of the members of the ICMR expert group, I had suggested to ICMR to collect all the sequences of the kit manufacturers who are producing kits in India and do a bioinformatic exercise and see how many genes are conserved in the conserved region of SARS-CoV that no mutations are reported, just to keep a watch on whether kits are likely to fail. And I must say, ICMR did a wonderful job. There's a huge database available at ICMR. Dr. Nivedita Gupta has done a great job of it. So if any of you have any doubts, you could always reach out to ICMR. Dr. Chaitali or Dr. Devjani, you want to add anything on this question? Okay. Sachin, next question, Sachin.
Yes, I think the second question is, about, from an anonymous attendee. He wants to know that if there is an ICMR issued guideline to use test only with dual gene targets, because as we have seen that rapid tests with single gene targets are being massively deployed at airports. So how risky you think it is, and what can ICMR and all of us do about it?
Well, I'll ask all the panelists to take this an important question. Ashok?
I think you are the best qualified to answer that.
Okay, my own take is, let's apply common sense first and then apply science. More the number of genes in a test being amplified, I think the chances of missing it are less. That's what common sense tells you. And I think it's up to manufacturers to now come up, and a good manufacturer should come up and upfront say, look, I have done the bioinformatics of my particular genes that I'm amplifying, and give a declaration saying that we are variant proof. I think that's the least manufacturers can do for a pandemic situation when so many cases are being reported. So these are my two. As far as I know, there is no ICMR guideline that says you should or you should not use a single gene or you should use a multiple gene. There is no guideline like that. Whether ICMR will take such a call, I don't know. It's up to ICMR. That's my response.
And also, I would like to add this, even though if it's a single gene target, none of the airport, I don't think so, they are testing the kit which is not ICMR approved. So though it's a single gene, double gene or triple gene, all the kits which are used in at airports or in the lab are ICMR approved. That's all.
Exactly.
Thank you, Dr. Ravi and the panelist. Okay, I think we can take one more question, and it's an important one, I feel. So the question is about, since COVID positive patient need to be quarantined, should genome sequencing be prioritized over diagnosis and treatment? And how will presence or absence of Omicron variant help us in treatment?
Dr. Ashok, Rattan, Dr. Chaitali and Devjani, I think
we've all agreed that yes, since they'll be isolated, especially it will be in the airport, they'll be isolated. So we must do a quick test first to see whether you suspect a variant, and then confirm the variant and keep the person in isolation, in quarantine till that time has come.
as far as treatment goes, I'd just like to add, treatment for Omicron is no different from treatment of any other SARS-CoV variant infection, including the parent one. So I don't think it has any implications for treatment. The main implication, as Professor Rattan very rightly said, is, trying to keep the person isolated so that it doesn't spread into the community. But eventually it will get there. So initially all this hype will be there, but eventually it will get there. Any other responses from?
Nothing, I guess it's covered.
Thank you very much. Is that the last question, Sachin?
Yes, Dr. Ravi. We can.
Thank you so much, panelists. My personal thanks and namaskar. Value your time and your expert inputs into the. Thank you very much.
Thank you.
I hand over to the organ.
Thanks, Sachin, and thanks again to all the speakers, Dr. Rattan, Dr. Ravi, Dr. Chaitali, and Dr. Devjani for putting up this insightful discussion. Moving forward to the next section. Moving on to the next session, which is a presentation on the topic Solutions for Express and High Throughput PCR Testing, which would be taken up by Mr. Pulkit Kheria from the Project Management Team at Tata MD. Over to you, Mr. Pulkit.
Thank you, Khushbu. Good afternoon to everybody. I want to thank our panelists for having such a great first session and giving us this time. And thank you all for taking out time on this Thursday afternoon to attend what we feel is a timely and important discussion. In this session, we would like to share with you two of solutions in our portfolio that we feel can have a significant impact in first-line testing for the coming Omicron wave. I will just share my screen. We've prepared a few slides. Please give me a minute. And before we get into these two solutions, I'd like to take a minute to talk to you about Tata Medical and Diagnostics, or Tata MD as we call it, our company. We are a venture from the Tata Group, which we've all come to so like and admire. Our vision, we are here to make healthcare more accessible, more affordable, and we believe that diagnostics that is decentralized, that is available to all, is absolutely a foundation stone for such a healthcare system. And that is why we have come into existence with the goal to bring diagnostic and medical care solutions and products to the market. We started off this journey with Tata MD-Check CRISPR, the first scalable CRISPR diagnostic in the world. And then since then have moved on to expand our portfolio to a full set of RT-PCR and antigen tests for COVID, as well as turnkey solutions for managing COVID. We're very much committed to solving the pandemic problem for India and for the world, and we will continue to support the healthcare community in tackling this issue. At the same time, we very much aspire to be a full-service diagnostic products company where we will have a portfolio, comprehensive portfolio that will span, of course, molecular diagnostics, but also immunoclinical chemistry, hematology, and as the technology evolves, whatever else can suit the needs of users in India and in global markets. So with that, coming back to the problem at hand, really COVID-19 testing, as we see it, poses some very unique challenges and pressures on the testing systems and the technologies that are deployed. And to meet those challenges, we've come up with two unique solutions. One on the left that you see, Tata MD-Check 𝐻𝐍 is an extraction-free fast PCR that works with our Express PCR analyzer. It is a specifically designed complete kit plus instrument solution that can help you process 30 samples at one go in as much as 60 minutes. And when we say 60 minutes, it is from sample collection to report generation. The other solution that we would like to talk to you about is our Rtpcr Fast 3-gene kit. This is a kit-only solution that is ideal for existing qPCR setups, where you have existing automated RNA extractors and qPCR machines. This solution can help you significantly cut down the TAT and improve the utilization of your setup. I'd like to take some time to deep dive into the 𝐻𝐍 solution with Express PCR. As I mentioned, this is a complete solution with an equipment and an extraction-free kit. The challenge with current rapid solutions, and this is broadly a challenge in our industry, we've had sort of two polarizing technologies. The point of care technologies have focused more on ease of use. They've focused more on sort of easy operation, fast turnaround time. On the other hand, the lab technologies have focused more on gold standard accuracy, scale, and high volumes. I think COVID has got us into the situation where the both need to come together. A COVID testing solution needs to have gold standard accuracy. It needs to have easy operation so that it can be deployed close to patient in places like airports, large events, special VIP gatherings, etc. And at the same time, it needs to be scalable. And that is where we think we bring a unique value proposition. The solution that we are offering allows you to process 30 samples at one go in each instrument. These instruments can be easily deployed in parallel, where 5, 6, 7 instruments can be operated from a single laptop by a single operator. That is where we also believe some of the existing rapid solutions are not able to do justice. You have to employ a battery of devices, each with its own interface, which requires huge manpower requirements and very complex operational workflows. Here, none of those issues will occur because all of these machines can be deployed together and operated by a single operator. The underlying platform, the underlying fundamental technology still relies on the tried and tested qPCR or real-time PCR technology. And so the learning curve is much shorter. The workflow is very simple. There is a sample processing buffer that is provided in the kit. The sample needs to be inactivated with that buffer for 5 minutes, and then added to the reaction mix and amplified. That is it. There is no extraction. The only extraction that is happening is liquid-based. There is no use of automated extractors, and the reaction mix itself is a single tube, one-step RT-PCR. Also, the fact that this platform is actually building upon the qPCR technology makes it much more extendable and useful even beyond COVID. Whatever technology we build now, it will be easier to build beyond COVID assays, leveraging a tried and tested technology like qPCR versus something like an isothermal amplification that might require specialized development and lot of de novo R&D. Ultimately, this kit, the next point that I would like to cover, it is a fully multiplex 3-gene kit with an internal control. Our panelists spoke earlier about how having multiple genes does improve the probability of us not missing any variants in the future. So this solution is going to offer gold standard accuracy variant-proof testing, whether it's Omicron, Delta, or any other variant of concern. None of those affect the assay. The next point that I'd like to discuss with you is the capability to collect samples in VTM. Here is where, again, some of the existing rapid solutions add complexity to the workflow, because many of them, in fact, most of them will use a lysating medium, and the lysated sample is not suitable for follow-on sequencing. So should a patient be found positive, the sample has to be recollected. That problem will not occur in the Tata MD-Check except Tata MD-Check Express PCR solution, because you can collect the sample in VTM and continue to send it for follow-on sequencing or for long-term storage. There is no stability issue. So these are some of the advantages that we see in our XF solution combined with our Express PCR analyzer. We believe it's the only scalable rapid testing solution that is available right now to address the needs that might arise should the Omicron wave hit us. And it is ideal for use at near patient crowded settings like airports, large events, offices, workplaces, factories, construction sites, etc. The second solution that we would like to talk to you about is something that is ideal for existing qPCR setups. Labs that have automated RNA extractors, qPCR machines, and are bracing up for an Omicron wave will find this solution very helpful. This is again a 3-gene plus internal control fully multiplex kit, but it comes with a 40-minute amplification protocol post-RNA extraction. This allows you to cut down a batch processing time by anywhere from 45 minutes to an hour based on whatever kit that you're using today. That is, of course, useful in offering faster turnaround times to the patients, but at the same time, once you are under the pressure of delivering a number of tests, something like this can help you increase the utilization of your existing capacity without having to invest in additional capital. So in that sense, the fast solution helps you service your patients better and at the same time realize better utilization of your existing infrastructure, create more impact to help us tackle the Omicron wave. That is the end of this session. And with this, I would like to hand over to the moderator with a final word that we all hope that the Omicron wave does not become as menacing as some of the previous waves that we have seen in the country and across the globe. And we sincerely hope that. But should that happen, these two solutions are our sincere effort to help the country and the globe prepare for this problem of COVID that we hope will be behind us soon. I'd like to hand over to the moderator, please. Thank you all very much for listening.
Thank you, Mr. Pulkit, for putting up this presentation together for our attendees.
Sure. Thank you, Kushbu.
Moving on to this discussion, we have Dr. Saurabh Arora with us. He is the Managing Director at Auriga Research Private Limited. So we'll be presenting on the topic, implementing fast and high throughput COVID testing at airports. I welcome Dr. Arora. Over to you, sir.
Hello, and a very good afternoon to all. I hope you can hear me. Hello, can you hear me?
Yes, we can hear you, Dr. Ravi.
Oh, that's great. So, I'm just sharing my presentation. So, I hope the presentation is also visible now. You will have to excuse me. I'm joining I'm actually in transit, so you'll have to excuse me for that. I thought when I was called in to talk about this presentation, I will cover specifically some peculiar challenges that we have in the scenario when we are working at the airport. Overall, for all of us, the entire fraternity, the COVID testing has been a great challenge involving scalability, rapid deployment, learning curve, and new technology and new challenges coming. But I'm actually taking this opportunity to talk more specifically on things that we feel are somewhat unique also to the airport environment. So first and foremost, turnaround time for all of us working in the labs, it has always been the one of the most important measures, right up there with the accuracy and the results being given to be true. But at the airport, there is a difference in that the expectation of turnaround time is talked in terms of minutes, not in terms of hours, and definitely not in days. So this has been a big change of the mindset and even the lingo in the lab with the team, that we are monitoring and implementing things with the view of cutting down minutes and improving the delivery. Why is this? Because when the lab slows down, there is a visible and huge impact on the passenger flow through the terminals. We must realize that the airports are not designed to keep people waiting for anything. They are designed for people to move through and out of the terminal very quickly. So when we introduce steps where we are forcing people to wait, it can lead to huge crowds and further create risks due to inability to follow the COVID measures which are needed in this current time. So we have to monitor and implement strategies to manage that very, very aggressively. So obviously in this, the conventional PCR workflow just cannot meet the requirements. We cannot have a lavish 90-minute PCR run and half an hour or 1 hour extraction. So we have to keep on looking for new things. Beyond the PCR workflow, we also have to work on optimizing the passenger registration and reporting, because this is all happening very real time, from the passenger landing to the registration and reporting. All of it has to be done within minutes, and every step adds. And in fact, when we are talking with the terminal team or the government, state government also, the TAT is actually calculated from the first passenger coming out of the aircraft to the last passenger leaving the terminal. So it is a whole new way of looking at it. Then after the different definition of TAT, there is also the issue of highly variable volumes. There are multiple factors which cause this. There are frequent changes in international regulations. Today, some country will say we need a rapid antigen test. Tomorrow, another country will say we need a rapid PCR within 6 hours. Somebody will say we need it 4 hours. Somebody will say 24 hours and all. So sometimes samples will go up, sometimes they will go down. There are also similarly changes in domestic regulations with how the disease is spreading and these new variants leading to increased monitoring and screening of passengers. And these are even now still on the macro level, but there are a lot of micro level fluctuations. Like every day, there are different loads of flights coming in, different number of passengers in each flight. So there is no steady stream of samples per se, and whatever system we use must be able to manage from a very few samples to a very large number of samples while maintaining the same turnaround time and accuracy. Because we don't have the luxury of waiting for 5-6 hours to collect samples, or even 4 hours to collect samples and then run a complete lot. So for this, we need to be able to run small batch sizes and take up samples as soon as they are collected. We should be able to run them. At the same time, we must be able to avoid any wastage, because if we go with the traditional system of the 96 plate and the pre-filled plates and all, which is very good for high throughput, large volume, it immediately becomes a very wasteful and expensive exercise. When you have departure flight, they also needed it to be done within 2 hours. Then, because of all these factors put together, it has to be highly scalable, almost instantly, because tonight something new will come and tomorrow the new regulation has to be implemented and it can lead to huge chaos. So the scalability should be not only in terms of the machine, but also being able to train the manpower and roll out the skilled people on that workflow, and lab space also needs to be accommodated. We also need huge amount of redundancy, because there is no chance for failure because we are working on such small turnaround times. So for redundancy, it has to be a simple workflow so that the manpower is there. Machines, we need to have multiple sets so that there is no single point failure. Then material supply chain also needs to be very robust so that irrespective of what is happening, we are able to get the kits, because it is very difficult to predict because the volumes are so variable. We can't say, okay, let's keep a month stock. We have to order stuff every day. So we have to deal with multiple vendors and very efficient sourcing. And lastly, again, the redundancy is not limited to the lab, but technology as well. All IT infrastructure being used for interfacing with the passenger needs to be very high availability, so that there is no, like a flight comes, 2 flights come, 600 people registering together. Still, you should not have any failures. So the technology part is equally important. So if I was to talk about my ideal workflow, keeping all the learnings from the last year in mind, so I would love to have small batch sizes, give results very fast with minimum sample handling, and something that needs very less space so that I can scale up. And lastly, we must be able to run multiple batches in parallel. So when we were talking to the team at Tata, so we actually identified that yes, it ticks a lot of these points, and we can have a 30 batch, or we can actually even run a single strip in the machine with just 15 samples in there. It is in strips, not in plates. It's a total about 50 minutes of runtime. There is no extraction, so less handling is there. Modular benchtop systems are there, so you can add more in less space. So here are some pictures of what we have together implemented at the airport, and hopefully we will be able to deliver very well with this infrastructure. So here you can see one of the sampling registration areas. In the background you can see the mobile lab. So this is also very important that Tata helped us increasing the lab capacity by providing this prefabricated mobile lab, which immediately added lab capacity for us. There are some pictures from inside the lab where you can see the 5 systems kept in a very small space. So I don't want to sound crude, but it's about the size of a pressure cooker. And you see 5 systems here connected to one laptop. So yeah, that's it from my side. Thank you so much for the opportunity, and it's nice to interact with everybody and the experts, all of us fighting this situation together. I hand over to the moderators, please.
Thank you, Dr. Saurabh. Really appreciate you sharing your insights with us from the real world experience that you've had, and also being so kind to what we've been able to do with Auriga. It's just the start of the journey, and we hope together we can make a large impact at the Bangalore airport. I'd like to invite Dr. Ravi to join us for the Q&A session, and would like to invite all our participants to please pose any questions regarding what Dr. Saurabh and us are trying at Bangalore Airport or achieving at Bangalore Airport, or more broadly about the solutions that we've talked about on this event. So request the participants to pose questions that they might have.
So while the questions are coming, I think there is a, I can see, some of the few questions around the PCR analyzer, the Express PCR analyzer, and, what has been, experience of using it, at the airport, and especially on the training requirements. I see that there is some few questions around that.
Dr. Saurabh, would you like to take that and share the experience of having the Auriga staff trained on the Express PCR analyzer from Tata MD?
In terms, like I said, in terms of the workflow, it is quite a simple workflow, and the instrument is a very familiar PCR kind of interface that most of the users are used to. So, and over and above that, we've had the Tata team here with us for supporting and for handholding and training our team. I think more than 15 days they've been here working day and night alongside the team. So wonderful support from their side. But yes, at the same time, it's quite the simple workflow which anybody who is used to setting up a PCR and running it can very easily do. There is nothing complicated in terms of even the programming of the system or whatever. It's a very simple workflow.
Thank you, Dr. Saurabh. There's a second question, which is probably Dr. Ravi, you or one of the panelists can take. It is from Dr. J. Gerard. He says that will CT value will correlate with the severity of the disease due to Omicron variant?
Dr. Ashok Rattan, you want to take that?
No information about the severity. Actually, from all accounts, Omicron seems to be very mild, mild infection. So, but of course, CT value in the past from New York, they had said it correlated with whether a person would survive or not. And I think there was a study from New York which said that if the CT value initial on admission CT value was more than 5 point something logs, then the person would not survive. But besides that, there's been no other study linking CT value with live infection, live recoverable bacteria, virus is there, but CT value with severity, only one study.
Thank you, Dr. Prav.
So there is a question from Sheila Praveen. She is inquiring about if the instruments, the analyzers are available in the market for standalone labs, and the cost of the instrument and cost of each test. Pulkit, do you want to take this up?
Sure, I can take that, Sachin. Yes, Ms. Sheila, the instruments are available for standalone labs as well. Regarding the cost of instrument and test, I would request that you can leave your contact here, or you can drop an inquiry on our contact email address, and our sales team will get in touch with you.
There is a question on what is the in terms of the utility between the Rtpcr Fast 3 gene and the Rtpcr XF. I think, Pulkit, you highlighted it in the presentation, but if you could repeat it for everyone's benefit, it would be very helpful.
Sorry, Sachin, could you repeat that? Did you say The XF versus the fast 3 gene?
Correct.
Okay, understood. So when we talk about XF, let's talk about it in the context of the full Express PCR solution, which is the analyzer and the kit. That solution is designed specifically for new capacity buildup that can initially tackle the menace of Omicron and then later can be extendable to other use cases. It is an extraction-free solution that comes with a kit that has been specially optimized for this analyzer. So in that sense, it is, for practical purposes, a full solution, a controlled solution, a semi-closed solution. And so that is regarding the Express PCR XF solution. On the other hand, the fast 3 gene is a kit-only solution. It is an open system and it will work on most widely available 4+ channel qPCR platforms. The 3 gene fast kit does require RNA extraction. It is not extraction free, so you will need manual or automated RNA extraction methods with that kit. And it is ideal for existing qPCR setups that can reduce the turnaround time and also increase capacity utilization. So, so just to summarize, Express PCR and XF for rapid PCR testing at scale, at airports, at large events, at gatherings by building up new flexible capacity. On the other hand, fast 3 gene for faster, more efficient testing in existing qPCR setups.
Thank you, Pulkit. And I see multiple questions on the cost of the kits and the instrument. So I would request everyone to, send the, the queries, either they can leave it at the chat and we'll get back, or they can send it on inquiry@tatamd.com. I'll put it in the chat window. Dr. Saurabh, there's a question, I think probably you will be the best to answer. There is a query on how long do you think these COVID testing for travel do you see in the foreseeable future?
I really wish I could answer that question. Even I don't know, because it would really help us plan and predict. But if you look at the unfortunate situation that is right now, I think maybe at least 2-3 months, and hopefully travel will continue. That is more important. I don't mind if testing continues, but travel must continue, and I hope that situation doesn't come that we have to again shut down travel. So all of us in the testing are happy to be here and support that objective, that we can continue to have travel and make it safer. But nobody can say till when this testing will do.
I think the intent of the question is to understand that whether the testing at the airport or travel testing, is it going to be phased out very soon, or is it going to be something that probably is going to be there for at least some time?
It is going to be there till I think at least for next 2-3 months, I don't see it stopping.
Thank you, Dr. Saurabh. There is a specific question from INR Pandian. He wants to know the gene sequence from MTM sample is also possible, because he's referring to certain articles saying it is possible with MTM and MTM samples as well. So may I request Dr. Ravi or one of the panelists to answer it?
Yeah, it is possible. It depends on the quality of the MTM. It has to maintain the integrity of RNA well. So if it is a tested, approved MTM, yes. But the disadvantage with MTM is you cannot culture the virus, because MTMs, as you know, lyse the virus. So if you're looking at culture and sequencing, then BTM is the solution. But yes, MTM can be used. Specimens in MTM can be used for sequencing.
Thank you, Dr. Ravi. I think a follow-up question is on whether we can do pooling testing as recommended by ICMR.
No, not for international travel. For international travelers, this pooling is not recommended. general guidelines of ICMR for pooling are still valid in the community settings when the prevalence is less than more than 5%, then pooling becomes very cumbersome. Less than 5%, it can be done, but there are many riders to it. I would advise to go to the ICMR website and look up the guidelines for pooling, but certainly not for international travelers.
Thank you, Dr. Ravi. I think we have covered majority of the questions, and for specific questions on product, I think I've already posted, an email, that you can refer to. So that will be really helpful. So thank you everyone for participating in it, for all the questions, and now I hand it over to the moderator.
Yes, thanks. And with this, we mark the conclusion of this webinar. Thanks again to all the speakers and the attendees for attending this webinar, and also to the speakers who have presented an informative set of presentation. Thank you, everyone. And I think we've concluded the session.